Right, so direct RNA sequencing of SARS-CoV-2. We were in a lucky position at the Doherty Institute in that some of our colleagues were the first outside of China to culture the virus. And so, they were happy to provide us, and particularly George, who led this sequencing effort, with material for direct RNA sequencing. And then there were 3 papers that came out actually quite soon after one another. We were the first, I guess, because of this early access. And so we could look into, I guess, what we could say about the genome of the virus. So just to set the scene, if you like, this genomic structure of SARS-CoV-2, it's just shy of 30,000 bases. And on the 5' end, you have the non-structural proteins, which 16 non-structural proteins, but they're encoded actually only in 2 open reading frames. And there's a translational slippage that occurs between the 2. And then the 16 proteins are then cleaved from the protein afterwards. And then on the 3' end, you've got your structural proteins, so the spike envelope, the M and the N proteins, and then your accessory factors as well. So obviously when you do sequencing, the first thing you look at is the coverage. And so this is what a characteristic, and it looks like this for all coronaviruses pretty much. So it's a polyadenylated RNA molecule. So and that makes it obviously easier to sequence using direct RNA sequencing. And so just focusing on the left, we sequence 2 different types of RNA from 2 different sources. The first is from the culture, so grown in the Viro cells. And so Viro is very permissive for growing SARS-CoV-2. And you can get lots of reads, as you can see here. And then on the right, we also sequence material from Virion, so not grown, so it's from the supernatant essentially. And it's much harder to extract RNA from there, and you get much lower coverage, but we can still get some interesting data out of it. And so you see in the light, the light color here, the light green color, that there's a very high depth on the 3' end, and it drops off, and then it picks up again a bit to the 5' end. And then, so that's all reads, but the way the SARS-CoV virus manages to translate its 3', so the translation obviously occurs 5 to 3, but it's quite a long virus, 30,000 bases. So in order to translate the 3', it has to do a bit of a trick, which is to use kind of a splicing-like process, so that the negative strand is continuous extension, whereby it joins up the 5' end, which contains a leader sequence which promotes translation, and it joins that up with the 3' end. And so that's the reason we are particularly interested in reads which have the 5' end. And so that's why we plot here the so-called leader reads. They're reads which map to both the 3' end, which is kind of by default because we're sequencing from poly-A tail, and then the 5' end. And so you see this is a reasonable proportion of the cellular data, but a much smaller proportion, in fact, of the Virion data set, I guess reflecting the fact that the Virion sample is not undergoing active formation of these characteristic subgenomic mRNA. Okay, so if you then take these leader-containing reads and look at the length distribution of them, you kind of see quite quickly the different characteristic subgenomic mRNA. So this is purely just the length distribution, and so you first see the ones which are close to the 3' end because they're shorter essentially because they've had the 5' end spliced on, and so they're much shorter. So you see this N nucleocapsid gene first. You also see that there's this ORF10, which I'll come back to, which seems to have nothing there. And we concluded in our preprint, in fact, that there was no ORF10 expression, as did the other 2 preprints, in fact. But I'll come back to that later and show that I actually do think there is some ORF10 expression, even though it's very little. And you can see the other open reading frames. And then, so the top is the cellular material, and then the bottom is the Virion material, and there's much lower coverage, but you can still see that in general, you get the same peaks. So this length-based clustering into subgenomes is kind of okay, but it obviously falls short if you have things which are quite close in length together. It's quite hard to cluster a histogram or reads from which are forming the histogram into the different clusters. So we actually wrote a new algorithm for doing that, which is fairly simple. It's based on clustering of the sgR string, if you like. So you first map all the reads to the genome, and then you inspect the sgR string of each read, essentially. And you look for the position of this junction where it's spliced the 5' end to the 3'. So that manifests itself in the sgR string as a very long sequence of ends, essentially. So you look for the longest sequence of ends to find the largest breakpoint. And after you've identified that, you find upstream, the open reading frame immediately upstream of the 5' and then the first start point of the open reading frame immediately downstream of the 3' breakpoint. Because the idea is that the translational machinery will start translating wherever it hits its first ORF downstream of the breakpoint. And if there's no upstream ORF, that just means it's a fully leader. It goes right to the start and has that sequence at the beginning which promotes translation. And then we just grouped into the pair of upstream and downstream ORFs. And in parallel, we would also calculate the usage of the breakpoints. So once you do that, the coverage plots begin to make a lot more sense. So this now is the coverage plots of the leader containing reads from the cellular material colored by their different cluster. And you can see that now there's very sharp, distinct breakpoints where the coverage drops to 0. So the NORF is the highest expressed open reading frame, and you see there's a point here at which the coverage drops to 0, and that occurs almost right at the start of the N gene, just a little bit upstream of that, and it coincides with a motif which is indicated in these dashed black lines, which is the so-called TRS or transcript regulating sequence motif. And so there's one you'll see right on the 5' end or leader sequence as well. So that controls where this splicing process occurs. And so you can see that each of these open reading frames has a very sharp breakpoint at which that occurs. You might notice on the left-hand side that there's this blue line which extends out a little bit, and I'll talk about this in a second. So we do see some subset of reads which have like an extended 5' sequence. So just zooming in, so this plot simply zooms in on the previous plot, both on the 5' end and also on the 3' end, so the 5' end on the left, but the colors match between the left and the right. So the middle bit is in some way less interesting because it gets spliced out. And so you see that there's the leader sequence, has a very strong break there as well. But there are these extended leader sequences kind of carrying out past 100 base pair and past 1,000 base pair, in fact. And then on the right-hand side, this is on the log y-axis, I should say, you see that many of these subgenomic mRNA do have the sharp breakpoints, but actually now we can look at some of the ones which are slightly less abundant. And in particular, I'll draw your attention to the purple line in the bottom right-hand corner. And this actually corresponds to reads for which the next downstream open reading frame is actually ORF10. And so this is the reason we missed it from the histogram clustering point of view, because there's not a sharp breakpoint. For the histogram clustering to work well, you really need that sharp breakpoint to have all the subgenomic reads having the same length. And in this case, there's kind of a gradual build-up, although it is biased towards the 3' end. You may also know some of these ORF10 reads actually have this extended leader 5' leader sequence as well. So as you can see, we can estimate from the depth the kind of level of coverage, and we can compare. We can do all those plots I showed you for work for the cellular material, but we can show the same information for the Virion, and even though there's less reads, we can see that, in general, the level of coverage for the Virion sample, correlates well with the cellular sample, I guess with the exception of the spike protein. But at that level, that's, very low number of reads, so that's definitely could be statistical noise. So just looking a bit more about the breakpoint analysis. So these plots are heat maps with a log intensity scale, indicating the read depth in that scale. And so red is the most abundant, and then grey means there's nothing there, and blue is the least abundant. And then on the up the top, the plots. So on the top of the columns and then on the left of the rows, I've indicated the open reading frames. And then the solid white lines within the heat map indicate the location of these transcript regulating motifs. And you see that there is a very strong preference on the, so I should say the y-axis is the 5' end and the x-axis is the 3' end. There's a very strong preference for this splicing event occurring at around 65 base pairs at the 5' end. And here you can also see that the most abundant one is just upstream of this N-ORF as well, reflecting the fact that this is the most abundantly expressed open reading frame. But there is a bit of wobble. So you do see that, it's not a completely perfect process. There is a bit of wobble in terms of the actual position at which it splices. And then on the right-hand side, we've zoomed out on the x-axis, sorry, on the y-axis. So we've zoomed out to, instead of on the left-hand side of the plot, we had 0 to 100. On the right-hand side, we've gone 0 to 1,000. And so we can now see some of these non-canonical splicing events, if you like, which extend further than on the 5' end than just the leader sequence. And you see there's quite a number of those. And that there's a number which seem to line up just directly in front of the ORF10 open reading frame as well. So those transcripts, in fact, would bring together part of the ORF1A material together with the ORF10 material in the same read. Now, as to whether it's the ORF10 material gets translated into protein, that may not be the case because it will probably encounter a stop codon before it gets to that point. But nevertheless, it's interesting that ORF10 is being added to many transcripts, and we speculate there may be some kind of transcript stability role involved in it. So the other thing we saw was we looked at the base calling error rates from direct RNA sequencing data. And so this is just showing the distribution of all reads which which belong to a particular cluster. And I guess the most striking thing to see here is that the Virion sample has a greater error rate than the cellular material. So we don't know yet, of course, whether that's because there's perhaps more RNA modifications on that sample. We could speculate that might be the case. However, so we also, one of the advantages of this kind of clustering approach is once you've clustered reads, you can then do consensus calling, and because you've got a consistent breakpoint for every read in that cluster, and so you can do consensus calling and then get a much better error rate. So this shows you the error rate that we can attain after we consensus call each of these open reading frames. And so we do see fairly dramatic improvements in many of these open reading frames, particularly ORF6, for example. The Virion, we get down to 0 error rate. Some of them still remain a little bit high, and so there's definitely room for improvements in the base calling accuracy. But certainly consensus calling is good. And after we've done consensus calling, we could also see, it's not indicated here, that in fact, the Virion sample has a 10 base pair insertion relative to the cellular material, or vice versa. The cellular material seems to have a 10 base pair deletion. And we can speculate as to why that might be the case. It's something we can see much more clearly after doing the consensus calling. So I will just conclude. So direct RNA sequencing is quite a nice tool for providing insights into transcriptional activity of SARS-CoV-2, in particular elucidating the subgenomic mRNA. I didn't show this, but you can also look at poly-A links, and you can start to look at putative RNA modifications as well. So we reported about 42 C modifications, but we haven't done any kind of experimental validation of those. So ORF10 is interesting. It actually has no known homology anywhere. So I don't think anyone really knows where it came from. And also it may be expressed, but if it is, it's very, very low levels. And so it's unclear what it does and why it's important, really, but it's certainly there. And there's certainly a lot of splicing events which happen just upstream of it, particularly with the extended leader on the 5' end. So there's much less subgenome mRNA expression in the Virion data, reflecting the fact that it's kind of not actively undergoing formation of the subgenomic mRNA. And also the Virion RNA base calling rates are a bit higher. So and all of the software we've developed is open source in this package too. So I'll just finish by acknowledging, especially the people who provided us with the material so quickly. So from the Victorian Infectious Disease Reference Laboratory, this project was really driven by George Taurara, who is in Deborah Williamson's group, and in particular Daniel Rawlinson did a lot of the analysis and development of the software packages. Thank you.